we have Taylor Heckman. She grew up in Hawaii, where she was able to spend way too much time hanging out with fish, Taylor, if that's even possible. She received her BA in biology from Willamette University in Oregon, splitting her time researching aquatic bacteria and Hawaiian monk seal health. Then she went on to her current placement at UC Davis in the Aquatic Animal Health Lab under Dr. Esteban Soto, where she is wrapping up her third year of her PhD program. Take it away, Taylor.
Awesome. Hello, everyone. I'd like to start off by thanking the organizers for putting this seminar together and thanking my co-authors for helping put this project together. So like Dr. Phelps said, I grew up in Hawaii, and like, how could you not fall in love with the ocean and its inhabitants when the water is that blue? And I was lucky enough to work with sharks and fish and seals for several years before I discovered that microbes are really cool too. So it made a lot of sense to combine those two passions in grad school. So I'm living in Davis now with my pup, who arguably loves water even more than I do. And my research centers on a pathogen called Streptococcus iniae, which many of you were probably familiar with. So it's a gram-positive bacteria. That's one of the causative agents of streptococcosis in fish. And what I found particularly interesting about this pathogen is its broad range, both in terms of geography and host types. So outbreaks, often with high mortality, occur in countries all over the world, in over 65 different species. And that includes cultured and wild fish. It includes fish from warm and cold waters and fresh, brackish, and marine systems. And it's also an opportunistic zoonotic pathogen, sporadically infecting humans and other mammals. So despite the importance and wide range of this bacteria, there's lots of gaps in our understanding of it, and crucially, in our ability to effectively control and prevent outbreaks. So current methods of controlling outbreaks rely heavily on antibiotic administration, but variable efficacies and concerns with health and antibiotic resistance make this an unsustainable long-term strategy. And vaccination efforts have also been met with limited success due to the variability of this bacteria and the limited ability of these vaccines to protect against diverse strains. There are a few commercial vaccines available for Strep iniae, and those currently used in aquaculture are restricted to killed whole cell preparations or bacterians. So those are generated by killing or inactivating a strain of bacteria by any means of, or a couple different methods, such as uses like a formaldehyde or beta-propiolactone. And these are stable and cheap with low risk for reversion to virulence or spreading in the environment. However, they don't stimulate a very strong immune response, which impacts the routes of immunization you can use. So often they require stressful injections with adjuvants to help stimulate that immune response, and later may require booster doses. So I was excited that Tim was presenting before me so he could demonstrate the benefits of a live attenuated vaccine, which is an alternative to these killed cell preparations, where live attenuated vaccines are a weakened version of the pathogen that doesn't cause disease, but still generates protective immunity, often to a similar level as the wild type strain. So they strongly induce both arms of immunity, and so require smaller doses that can be administered through more natural routes with less need for adjuvants and boosters. And so it's less stressful and less time consuming and labor intensive for the aquaculture workers. And there's concern. So the main concern with these live attenuated vaccines is the theoretical possibility that the bacteria could revert to its virulent state. But live attenuated vaccines have been used successfully both in animal and human medicine. For example, with the Edwardsella ictaluri LAV in catfish, and if you got the measles vaccine for people. Because LAV are essentially a living replicating version of the bacteria that can't cause disease and generate a similar immune response, that can be especially beneficial when dealing with iniae, as the killed cell preparations have been limited in their ability to protect against more than the strain they were prepared from, right? So iniae is a really diverse species, and that lack of cross protection has resulted in vaccine escape and reinfection of vaccinated stocks. In a previous study, I used multi-locus sequence analysis to genotype diverse isolates and found there were 4 major clades of Strep iniae impacting cultured fish in the North American continent, shown in the highlighted regions, clades A, B, D, and E. And in addition to being genetically distinct, these clades also differed in important phenotypic characteristics such as virulence, hemolytic activity, and extent of capsulation. Capsulation extent is particularly important as variation in the amount of capsule produced by these strains has been implicated as largely responsible for vaccine escape. So in order for a vaccine to be effective long-term, it needs to be protective against these diverse endemic strains and prevent that, and try to prevent that possibility of a new strain taking over. So the objective of this project, therefore, was to develop an effective live attenuated vaccine against Strep iniae with the specific aims of generating LAV candidates for each of the genogroups, assessing their attenuation by in vitro assays, and then moving selected candidates on to in vivo trials, assessing virulence in Tilapia, as well as antibody response and cross-reactivity. And then finally, immunoprotection by the best candidate strains. Out of the strains characterized in that MLSA project, 3 were selected, isolated from 3 economically relevant host types: LSU 01105 from Nile tilapia, Eco 8617 from rose spotted snapper, and WS10A from white sturgeon. And those sturgeon and tilapia isolates are especially relevant for aquaculture in the US and more specifically for California, where iniae has been recently causing outbreaks with high mortality in our white sturgeon aquaculture industry. You'll notice there were 4 endemic clades and I chose 3 isolates. That's because clade A was also consisted of isolates from tilapia with 99% sequence similarity to clade E isolates, as well as no significant differences in virulence or phenotype. So a clade E isolate was chosen to represent strains causing disease in tilapia. To generate an LAV, there are a couple different ways you can use to attenuate. So you can have targeted mutagenesis, where you are specifically mutating a virulence gene, or you can use random mutagenesis and screen isolates for attenuated virulence. And despite the specificity of the targeted approach, public perception of GMOs have limited application of LAV generated by this route. So random mutagenesis was chosen. And there are also several different ways to generate random mutations, one of which is passaging that bacteria many times, either in just some sort of growth condition like a cell line or just media. But a variant of this is to add a stressor, like an antibiotic, to enhance that mutation rate. So we used a Rifamycin antibiotic, which functions to inhibit the beta subunit of RNA polymerase and block transcription. And passaging in Rifamycin has been used to generate successful LAV in other bacterial species, including that commercial vaccine for Edwardsiella ictolari in catfish. And when I looked at the sensitivity of iniae to Rifamycin, the MIC was very low, only 0.312 micrograms per ml, showing the suitability of this antibiotic for our objective. So to generate those LAV candidate strains, I started 3 lineages from each of the 3 parent strains for a total of 9 different lineages. And these were passaged in increasing concentrations of Rifamycin SV sodium salt from the starting MIC of 0.312 to a final concentration of 250 micrograms per ml. And resistant colonies at each of these concentration points were cryofrozen for further analysis. The 25 microgram and 250 microgram points were selected for initial testing. Changes in virulence were first assessed by several in vitro assays, including growth rate, buoyant density, cytotoxicity to tilapia cells, and survival in whole blood. To measure that growth rate in liquid media, colonies were restreaked from freezer stocks and suspended in PBS before being diluted into brain heart infusion broth, aliquated into a 96-well plate, and then incubated with the OD 600 being read every hour for 96 hours. So you can see from these growth curves that there were demonstrated differences in the growth rates for several of the passage lineages. And this isn't really that surprising because previous studies have reported that acquisition of mutations that lead to resistance to rifamycin have negatively impacted growth rate. But it does have potential implication or impacts on some of the other in vitro assays that could have effects from cellular growth rates in a given time period. To assess whether extent of capsulation was affected by any of the accumulated mutations, I used a buoyant density assay, where essentially you layer a concentrated suspension of the bacteria onto a gradient and then separate it out by centrifugation. And where that cell layer ends up is or has been demonstrated to be indicative of how much capsule a strain has. So the farther it travels down the gradient, the less buoyant or encapsulated it is. So further travel means less capsulation. The passage strains did not obviously deviate from the phenotype of their wild type parents, but it's good to note that the LSU strains in blue were more buoyant than either the eco or the white sturgeon strains, indicating they're more capsulated, while the others likely show sort of an intermediate phenotype. So to test the ability of the iniae passage strains then to kill or damage endothelial cells, I used the Mozambique tilapia immortalized bulbous arteriosus cell line, or Tmbs, and challenged them with the different bacterial isolates at 30 degrees Celsius. I used a colorimetric assay that measures cellular damage quantitatively by measuring the release of a eukaryotic cytosolic enzyme called lactate dehydrogenase. This cell line was chosen because the heart is a target organ in some iniae infections. For example, this image here is taken from a fish that was infected with the ECO86 wild type strain. And the cytotoxicity itself is measured by comparing the percent of lactate dehydrogenase released in respect to a positive control. So none of the strains are hugely cytotoxic towards Tmbs, and there were no significant differences in cytotoxicity between the parent and the passage strains. You'll notice that the white sturgeon strains did not demonstrate any appreciable cytotoxicity by this method, and that's not unprecedented, as none of the tested isolates from that genetic group have ever displayed cytotoxicity by this method. SNEA needs to disseminate from the initial infection site in the bloodstream in order to reach target organs like that heart and cause disease. So the ability of the passaged isolates to survive in whole blood and evade the immune factors present was determined by collecting fresh heparinized blood from healthy adult tilapia and then inoculating that aliquots of that blood with each of the different strains. So the suspensions are incubated for an hour at 30 degrees before they are spot plated, and the resulting colony counts are compared to the initial inoculum in PBS. So here we have more of an interesting result. We see that survival of the passaged isolates in comparison to their wild type parents demonstrated some significant differences. So in at least one passage strain per clade, we had significant attenuation in the ability to survive the immune factors present in whole blood. So all of the ECO86 derivatives were significantly attenuated compared to their wild type parent, whereas we had at least one 250 microgram isolate from each of the other clades. And survival in whole blood has been previously demonstrated to be a better predictor of SNEA virulence in a host than cytotoxicity. So based off of these results, we selected the 200 microgram point isolates for further testing in vivo virulence assays. The in vivo challenge sort of had two objectives. One was to assess that virulence of the attenuated isolates in a whole living organism. And the second was to see if there was stimulus of the immune system against those LAV candidates. So fish were challenged with the wild type attenuated strains or sterile PBS as a negative control, and then monitored for 21 days. Dead and moribund fish were collected and sampled for bacterial reisolation and for gross and histological assessment. At the end of the challenge, blood was collected from survivors and serum separated out for future use in ELISA to look at that antibody generation. There are some interesting differences observed in the clinical signs between the control and the wild type and some of the attenuated strains. So here are our control fish. I fed them maybe a couple minutes before this and they've already cleared up all the food. They're happy moving around. This is the wild type, or a fish injected with the wild type strain of LSU 01105. And so you can see that they're lethargic, they haven't eaten their food, and you might have noted that there's was unilateral exophthalmia and opaqueness in the one eye of that first fish. In the one of the passage at the surface in a second. They also had some exophthalmia, but they survived much longer if like they would have survived longer if I didn't euthanize them after appearance of these clinical signs. So even though it was the wild type LSU strain was extremely virulent as expected, and there was actually only one fish by the end of the challenge from that parent. Still showed mortality and those clinical signs as noted, but interestingly, LSU 01105A did not kill any fish. So that was a promising sign. We also didn't have mortality in any of the sturgeon isolates beyond the first day, and there wasn't any re-isolation of bacteria, so that was likely a result of the injection from that challenge. Whereas there was one death in the wild type ECO86 strain, and bacteria was recovered from that fish. I then went on to look at whether or not the fish challenged with these isolates had generated specific or cross-protective antibodies or cross-reactive antibodies against the different bacterial wild type strains. So I used a method of indirect ELISA. For those of you who aren't familiar, basically you coat the bottom of your with your bacterial strain of interval, and then add a secondary and a conjugate antibody that allows you to have a colorimetric way of measuring the levels of antibodies generated against these different bacteria. So some cool results. I haven't had the chance to test the serum against the white sturgeon wild type isolate as I was going order of most virulent large. But some interesting things to note is that the antibody levels that recognize the ECO86 wild type are overall higher than that against the LS. That might have something to do with those capsulation level surface proteins on the bacteria. So that might be the reason why we have for the less encapsulated. So there are apparently other antigens that can be recognized in this on these bacteria. And again, that LSU A strain here, even though it didn't kill any fish, it still generated levels of antibodies that were comparable to the wild type, which is a promising result for the next steps. This is an ongoing project, so I need to finish up the ELISA, and then I can pick the best candidate strains for protectivity assays. Pilot study with IP injection type strains. And if I find interesting live attenuated vaccine candidates from those trials, I'll do some whole genome sequence analysis to determine what's actually changed, what's mutated between the parent strain and the attenuated one that's resulted in that reduced virulence. And I'd like to say thank you to our collaborators and those who who provided bacterial isolates, as well as my funding sources and my amazing lab. And I will take any questions, any comments. Like I said, this is still a progressing project, so I'd love input and feedback.
Great, thank you very much, Taylor. That was an awesome presentation. A little choppy there at the end, so if anybody needs some clarification, you can go ahead and unmute yourself right now or put it in the chat box and I can read it.
Hey Taylor, it was a nice presentation. This is Jan. How's it going?
Hi.
How many passages, I think I might have missed it, how many passages do you do to attenuate that bacteria initially?
Right, so I started out, I believe it was around 20 passages. So I started out in increments of around 5 micrograms. And then as they got more and more resistant, I increased that up to jumps of 25 to 50 micrograms per ml.
Okay. And you're using an antibiotic to help attenuate it as well. And I'm not really familiar with these kind of studies, but is there any kind of risk of generating or selecting for antibiotic-resistant bacteria right off the bat that you'll be using as a vaccine? So if reversion ever does happen, then you're going to have an antibiotic-resistant strain out there?
Right. So luckily, rifamycin isn't used to treat iniae. It is a issue for some of the, so rifamycin is generally used in medicine for things like tuberculosis. So it's used in a cocktail. And because it's so common to develop resistance against it, that's why it's not usually used alone ever to try to treat diseases. So hopefully that wouldn't be an issue because the main antibiotics used to treat iniae are oxytetracycline and fluorophenocal, which have different methods.
Great, that makes sense.
Yeah.
And then when you have an attenuated vaccine, is there a way of measuring the risk of if it will revert? Or do you guys do a sequence analysis or see where those mutations are before you market any kind of vaccine like this, just to understand the reversion?
Right, yeah. So the reversion rate definitely is what we could maybe do is look at just the overall mutation rate of the bacteria. But generally it's a pretty rare occurrence, not to say that it never happens. But definitely before you commercialize it, there's a lot of sort of considerations in both like how well it might spread in the environment, if it poses any risks to different organisms, etc., that we'd want to follow up with. And mutation rate might be one of those.
All right, great, thanks. Sorry, I loaded that one with 3 questions. So
oh, no worries.
So I have a question. This is Stephen Fertini.
Go for it.
So I like that we had these 2 talks together, and I'm very interested in the some of that upfront work that you guys were doing with the bacteria. You know, the buoyancy and using the cell lines to try and look at the pathogenicity, or the virulence, I should say, of the bacteria. And then you went on and did the infectivity trials. And getting to the question, I've asked this before, that very often in the fish space, we just go directly to a survival study with some of these pathogens, and we're using a lot of fish just to get that data. Now I understand that you get different information from some of the tests you were doing that that can be used to interrogate the pathogens as they transform more deeply. But I'd be interested in your thoughts as to whether or not you feel that that type of work, that type of upfront work, though it may be a little more complicated, may help us start replacing some of this, go-to survival study work that we do. Right.
Yeah, I would say it can't 100% replace like a whole living organism, but like that survival and blood assay has been shown to be like in previous studies, even in for sure. They showed that it was a really good indicator of the actual virulence that would result in a live animal. And it was super useful for this project, especially because I had so many different candidates off the bat, because, at least 20 passages and then 9 different lineages, and testing all of those in fish would have been a nightmare. But if you have a more specific vaccine, I would say it helps put the whole picture together, and it can give you an idea to maybe save some fish lives off the bat if you want to reduce animal numbers maybe. So I think it's a good idea, but I guess it depends on, like, if you want to get the most, looking for a word there, the most representative results, right, which would be the whole animal trials, or if you want to try to have some predictive things first if you're
Mhm.
nervous about whether or not it's going to work.
Yeah.
How sure you are.
Yeah, thank you. That's the first time I've seen those. I haven't seen those really, talked about before.
Yeah.
Thank you.
Mm-hmm.
Well, that's all the time we have for today, folks. Taylor, that was an excellent presentation, and you too, Tim. Thank you very much for volunteering to be part of the online seminar. So a virtual round of applause for both of you before we take off for today. And just a quick reminder that we've got 2 more talks next week on viruses, so be sure to tune in to hear both of those. And have a great weekend, everybody. Bye.